and today I have come up with this topic of DNA sequencing. It is like one of the backbones or the crux of whatever you're going to learn in the coming days. You take a piece of DNA from the library, you have got many pieces of the single genome, right? So this is your single genome. You have many pieces stored up, many copies of this stored up in library. You take one piece and then you find out what is the individual base pair present there. So why? So sequencing means finding the order of nucleotides on a piece of DNA that we have established. Simple to understand. Nucleotide order determines amino acid order and by extension protein structure and function. What is this saying? Nothing. If you know what is the nucleotide order, from DNA you get what? You get the RNA, right? From RNA you get the amino acid sequence. Amino acid to put together is nothing but protein. So if you know what is DNA, you'll be able to know what protein is being produced by that. That is what is mentioned in this. An alteration in a DNA sequence can lead to an altered or non-functional protein and hence to a harmful effect in a plant or animal. Serious diseases. I have put up an example in the coming slide. You will see that. Now, understanding a particular DNA sequence can shed light on a genetic condition and offer hope for eventual development of treatment. So that's why, these are all the significance of why we need to do sequencing. DNA technology is also extended to environmental, agricultural, and forensic applications. So this is the example I was talking about. Okay, so DNA sequence variation can change the protein produced by a particular gene. Now, suppose this is your normal DNA sequence. Okay, this is coming over here like this. So these are codons, right? Triplet codes. These will, after transcription and translation, will give you an amino acid valine, then histidine, then leucine, threonine, proline. Then this one forms glutamic acid, and so on. The same thing, you have glutamic acid again. And this will give us a particular protein. This will result in having normal red blood cells in our bodies. But what if there is some mutation occurring? And instead of T over here in your DNA sequence, suppose what comes is A, adenine comes over here instead of thymine. So if adenine comes over here, the opposite base pairing will be with thymine. Nothing has happened. This has just flipped from here to here. Okay, one of them has mutated and the resulting one has again changed according to it. What is happening? All these will come be transcribed in the same way, but this, instead of glutamic acid, you have a valine. Okay, rest everything is same. You get a mutant. What happens from that single change in your DNA? You get something called sickled red blood cells, and you get something called a disease called sickle cell anemia. Imagine just one nucleotide change can cause so much change, really. So the change in amino acid sequence causes hemoglobin molecules to crystallize when oxygen levels in the blood are low. As a result, red blood cells sickle. Sickle means it's a uh, weapon. Okay, it's a long thin rod, not rod, it has sharp ends. It kind of looks like this sickle. You can dig with that. Okay, so that is why this, since your red blood cells shape becomes like a sickle, it gets that name. Okay, what will happen if your red blood cells shape will become like this? The oxygen that it can take in will become less, so you will suffer from something called sickle cell anemia. The blood oxygen level in your blood will be reduced to a large point. So I hope you're understanding what is the significance of having a proper DNA sequencing. So people are having doubt, what is causing this disease in body? So sometimes they will go for genome sequencing. Oh, so this is the normal human DNA. This is the DNA of a person who is having some disease. Let us see where the change has come. So for everything that you need to do, you need to understand what is the sequence. So C comes after A, comes after C, comes, so which is coming next. So that is why sequencing is so important. Now, how are genes sequenced? The first DNA sequencing project took 3 years to sequence 12 base pairs of DNA. Do you understand 12? Only 12 of them to understand what comes after what comes after what. They took 3 years. First start பண்ணும்போது. Okay, not now. Nowadays, it's like it happens very quickly. But the first DNA sequencing, it took 3 years. It was completed in 1970. Okay. So in the mid 70s, ladder sequencing strategies were developed. independently by Maxim and Gilbert and by Frederick Sanger. Now, these two group of peoples, okay, one is Frederick Sanger, and these people worked as a group, Maxim and Gilbert. These names, keep it in your mind. These people both gave two different methods of sequencing DNA. Out of that, the Sanger sequencing method, that is the one given by Frederick Sanger, is the one that is most commonly used today. Chain termination method by Sanger, or it is also called Dideoxy method, and the chemical degradation method by Maxim and Gilbert. And just a quick comparison between the two methods. So chain termination method you have over here, chemical degradation method here. So chain termination method, there are two ways you can do it. You can do it manually, but it can also be automated. Okay, so easier automated sequencing can be done. Whereas chemical degradation method, it cannot be automated and it is very time consuming. In chain termination method, you have no toxic substances involved, okay, or the toxicity level of chemicals used is very less. Whereas in chemical degradation, with the name itself, there is chemical. Many toxic chemicals are used over here, okay. Chain termination method, you sequence a single stranded DNA. Okay, and here you sequence a double-stranded DNA. This is just a basic difference, but I'll be telling you the advantages and disadvantages of both these methods in the coming slides once we cover up the methods. Okay, we'll go to the next slide. So, all of you know DNA is made up of polynucleotides, right? Nucleotides come together and they form a chain like this. Okay. So I told you in the first class what is a nucleotide made up of. It is made up of three things. You have a nitrogenous base. Okay. That is, it could be A, T, C, or G. That is one. Okay. Second thing, you have a sugar molecule over here. So mostly in DNA, you have the deoxyribose sugar. Okay, that is the second thing. Third thing you have is the phosphate group, right? So these are the three things that form a nucleotide. So now, how do these nucleotides form a bond? How do they come together and form a chain? So the reaction that occurs is in DNA, you have a hydroxyl group or OH group over here, right? Plus another nucleotide when it comes, this phosphate group is there, no? These two will come and combine with the release of a H2O molecule, that is water molecule. Okay, so this is coming from here, this is coming from here. With the release of an H2O molecule, only O will be left. Out of this OH and this OH, these both hydroxyl group, one O will be left and H2O will be removed off, and they will form a something called a phosphodiester bond. Okay, so that is how one nucleotide gets combined with another. So what you're seeing over here, I have told you that the ribose sugar has 5 carbons, right? 1, 2, 3, 4, and 5. So this is called the first carbon, this is second, third, fourth, and fifth. So you have a hydroxyl group at the 3' carbon. Okay. So the 3' carbons hydroxyl and the 5' carbons, that is, this is the 5' carbon, right? 1, 2, 3, 4, 5. The 5' carbon has a phosphate group. So the 3' carbons hydroxyl joins with the phosphate group of 5' carbon and forms a phosphodiester bond. I hope that 3' and 5' thing is clear because we need to know about it, okay, in the later slides. So it has formed a phosphodiester bond. The 3' carbon hydroxyl, the 3' hydroxyl present, and the phosphate group present in the 5' carbon. This is the numbering 1, 2, 3, 4, 5. Okay, with the release of water. So again, I'm just showing you the mechanism of DNA polymerization. Polymerization means you have different monomer units. Monomer is single unit. When they come together, two join together, you have a dimer. When many join together, you have a polymer. So that is a polymerization reaction. So this is your existing DNA chain. Suppose in the reaction you are providing something called a NTP. NTP is nothing but nucleotide triphosphate. 1, 2, 3. 3 phosphate groups. So in Sanger sequencing, basically in test tubes now, we will be adding NTPs. That is nucleotide triphosphate. So same reaction will occur. Okay, this phosphate group and this hydroxyl group will form a bond. With the release of this phosphate, this diphosphate group will be released off, and you will form a bond over here. So DNA polymerase is the enzyme which converts or catalyzes this. So DNA polymerase catalyzes the nucleophilic attack of the 3-OH on a phosphoanhydride. Okay, so you form a phosphodiester bond over here with this release. So that is how you will be forming the chain in Sanger sequencing. Now, before we actually get into dideoxy chain termination Sanger method of DNA sequencing, we need to know what is dideoxy. So as Rubika had told us, the sugar present in DNA is deoxyribose, correct? Now we have something called ribose, we have something called deoxyribose, we have something called dideoxyribose. So that is what I have put over here. So if your nucleotide triphosphate has a ribonucleoside, it will be just written as NTP. Okay, so that means what? It has OH group over here in the second carbon. In the third carbon also it has OH group, hydroxyl group. But by any case, if this OH, that is hydroxyl group, is replaced by hydrogen, that is that O is removed, what will we get? Deoxy. Deoxy means removal of oxygen. Okay, so we get a deoxyribonucleoside triphosphate. So that is denoted by small d NTP. I hope that is clear. And what if you remove the hydroxyl group from the 3' carbon and replace it by hydrogen group? Then you have what? 2 times oxygen is removed. 2 is di, right? So di-deoxyribonucleoside triphosphate. So that is what we represent by DDNTP. So you have NTP with the normal hydroxide groups. You have DNTP, that is deoxyribonucleoside triphosphate. Now you should know, in our actual DNA, we have this molecule, okay? We have deoxyribonucleoside phosphate. Okay, so this has an H over here and an OH over in the third 3' end. Okay, but if you modify this, you get something called DDNTP. And one more thing, this ribonucleoside will be present in what? RNAs. That is why it is called ribonucleic acid, right? So this, if ribo sugar is present without the oxygen, without the hydroxyl group being removed, this will give you an RNA. Okay, but if you have one deoxyribose over here, that means it will be giving us DNA. Now what will DDNTP do? What is its use? So dideoxynucleotides. So hydroxyl group on the 3' carbon of deoxyribose is replaced by hydrogen. Hence, it is called dideoxy. Now, this is incapable of being extended by DNA polymerase, which results in chain termination. I'll explain to you it in the next slide, okay? So, sequencing mixture contains all DNTPs and one or more DDNTPs. Random incorporation of DDNTPs halts DNA synthesis, and for given template, okay, this if I read out to you right now, you will not understand. I'll explain it with the figure. Okay. Now, suppose imagine this is your template strand. So whichever DNA you're going to sequence it, no? First of all, DNA is double helix, right? There are 2 strands in DNA and they are coiled together. Now what you will do is you will denature it. Denature it means you separate these strands and you will take only one strand. Okay. Out of the 2 stranded helix, you will denature it and take only one strand. Because if you can find out the sequence in one strand, you know what will be the opposite. It will be always complementary. So if there is A over here, of course it will be paired with a T in the next one. So if I just need to sequence one strand to understand what is there on the next strand, correct? So I take one strand, that is called the template strand. So in this diagram, this line shows a template strand. They have not drawn this over here, okay? They've just represented it with a line. Just understand that they have shown a line over here as the template strand. Now what they're saying is, if in the template strand there is a thymine over here, what will come and join here? Adenine. So your adenine molecule is coming and joining. This is your ATP that is coming and joining. Okay, A has come and joined. Now next base pair is C or cytosine. So which will come and join? Guanine will come and join. So your nucleotide with guanine has come and joined. Now next one is again G. G ko opposite lena join cytosine. So your CTP is coming. So this is the one that is showing the CTP coming and joining. So what will happen? This is coming, this will break over here, this will join with the 3' carbon. Okay, and C will join. Now, what if due to something, some reaction mistake, instead of a DNTP, a DDNTP comes and joins? That means ingame H ingame H. There is no OH over here. What will happen when this next CTP molecule will come? No, it will not be able to form a bond. Your chain will be terminated. Okay, it will not be able to form a chain. So that is the function of dideoxynucleotide, and that is why this method is called chain termination method, because in our 4 tubes that we take, no, we will be taking many molecules of DNTPs and also few molecules of DDNTPs. So randomly these DDNTPs sometimes will come and join, and once these DDNTPs come and join, the next molecule will not be able to form a bond here, and your chain will be terminated. Because I had shown you in the first figure, right? You need an OH group over here. In the 3' carbon, you need an OH group so that this OH group of the phosphate group comes and forms a bond. Okay, that is essential. If this OH group is not there and only H is there, this will not be able to come and join over here, and your chain will be terminated. This phosphodiester bond will not be formed. So that is the difference that DD-NTP makes. Okay, so the synthesis is stopped over here. So I hope you understand why dideoxynucleotide is being used over here. So we are learning about manual dideoxy method, right? So as I told you, now we are going to see step by step what we are doing. Okay.